Hello everyone, I am Sandeep. I am the Senior Global IVD Product Director for Fapon Biotech. Today we will be discussing about how Fapon's high performance diagnostic solutions can empower the local IVD manufacturers. Fapon as a company started like 22 years back when we were developing the raw materials and become one of the leaders in the core raw material for the infectious disease. Subsequently, we ventured into the core raw materials and the reagent solutions. So we've started focusing on the applications and became one of the leading suppliers of the raw materials for the emerging markets. Later on, from 2017 onwards, we started providing the one-step solutions where we have the core raw material reagents together with the open instruments. And from the last 3 years, we have the ecosystem cooperation where together with our one-stop solution and the conventional products, we are also looking into the ecosystem cooperation and the innovative incubation. This is driving us towards the industry innovation where we have global partnership in different particular areas. And we are continuously innovating and exploring the new technologies and the new products. Fapon has more than 1,400 products, around 800 patents, and we have more than 2,500 global business partners. We are a pioneer in the IVD technologies. So we have the high performance technologies for the generation of the antibodies. And we know a lot about the antibody engineering and most of the complementary technologies in freeze drying, how to improve the performance of the reagents. So this is something that we are doing routinely. With regard to the IVD one-stop solution, what is the IVD one-stop solution? You can compare it with the kitchen maker, which you are using every day at home. The kitchen maker can use the different types of the ingredients. And then you follow a particular program to make the different dishes. If your ingredients are always the same, you follow the same program, then you always end up in the same quality of the dishes. And it is exactly how the IVD industry is working. So we are providing the open analyzers, which are your kitchen maker. Then we provide you with the raw material and the reagents, which you are using within the analyzer and follow a particular program. And the stage-wise, the design phases for the development. Then you always end up in the same high quality of the IVD kits. And you follow the regulations ISO 13485, IVDR or the FDA 2030, so that you fall within the regulatory compliance. So we are providing all these solutions to the IVD manufacturers so that they can develop their IVD kits really quickly. Then to enable the IVD manufacturers to develop their products fast, we are helping them at every stage of the development, whether it is the initial phase where they require some customization of their reagents, screening of the blockers, customization of analyzer or raw material, or at the evaluation of the particular assays or the analyzer or the reagents. We also assist them in terms of the clinical evaluation. And then we share with them the production or the quality control SOPs and help them in the different types of the products, how they can develop, how IVD manufacturers can develop the products faster. For the after sales, we are guiding them step by step, whether for the installation, the software update or the training in terms of the users or the developer training for the analyzer. So we are assisting them on a routine basis. Now there is an extensive need for the open instruments. When we ventured inside the open CLIA analyzers, we realized the industry will change because automated immunoassays are dominating the market. So manual assays, which are being used by the small and the medium players, they may not occupy a significant share of the market. And these companies may not have the resources and everything that it takes to develop their own analyzer because it is very costly and requires a lot of skills. And all the big players are providing the closed analyzers. So we ventured inside the space and started providing the IVD industry with the open CLIA analyzers. which the clinical community can use for the development of new assays for the new biomarkers. And this will help them like in the discovery of the new biomarkers, and the new assays can come to the market very quickly. So in a way, we are increasing the sustainability of the SMEs so that they can develop their products very quickly. And we are creating the open IVD ecosystem. So we are democratizing the use of the open instruments so that the community can develop the assays really quickly. And we believe like this will lead to many novel and breakthrough diagnostic applications and the products in the near future. For the analyzers, our Shine Eye 1000 is the smallest footprint in the entire industry. It can do up to 120 tests per hour, and you can run 10 different types of the test with 30 sample positions. Similarly, Shine Eye 2000 is the medium throughput, 200 tests per hour, 25 different assays, and with 60 samples. And the largest one, Shine Eye 8000, 9000, is a modular system. So single module can run around 900 tests per hour. If you combine 4 of the modules, it can run 3600 tests per hour. And this is the highest in terms of the throughput for the entire IVD industry. And within a single module, you can run up to 410 samples and 60 different assays. So there are many proprietary technologies that goes inside the analyzer that I will discuss in the subsequent slides. And we recommend our customers to go for the actinium ester based flash chemiluminescence. But technically, we can also provide you the same system instead of the flash chemiluminescence in terms of the glow chemiluminescence also. Like actinium ester, ABEI, or for the flash chemiluminescence, and alkaline phosphatase and HRP, you are complying with the glow. So regarding the distinct features like of these particular analyzers, you can refer to our another webinar where we have gone too much in depth about the open clear analyzers. So some of the distinct features are their open system, they are compact, and they are highly affordable. We have the IVDR, and we are also going like for the FDA very soon, and they are fully automated random access analyzers. So the software is easy to use. You can go for the flash or you can go for the glow chemiluminescence as your signal output. And you don't require like a external PC because the system comes like with an integrated touchscreen PC, and you don't have to use like the stacks of the assay cups. This one is a single assay cup and you don't require any alignment. So you just put the cups in a particular containment and the system will do everything by itself. And the analyzers have non-touch vortex mixing, 3-phase magnetic separation for the washing. The washing is done in a unique way. So you have a thorough washing like of the dispensing needle without any carryover. And we are using like the high quantum yield hydrophilic actinium ester as a label, which has much higher chemiluminescence output. And the assay cartridge is the same for all of our analyzers. The reagents are refrigerated at 2 to 8 degrees centigrade. And the dispensing needle is made up of SUS 304 stainless steel, which is inert, heat resistance, and has high strength. So the analyzers can communicate to the laboratory information system via TCP/IP protocol. And as we already discussed, Fapon is a one-step solution provider. So we are assisting you like with all the services that you can think of to help you develop the IVD kits really quickly. Then you have access to our broad range of the customization services and the cooperation models in which we will empower you like to develop high performance diagnostics. And one of the most significant features is almost all our CLIA assays, they are biotin interference free. In 2017, FDA issued a safety alert mentioning like biotin as a supplement can interfere with many assays, and this was subsequently emphasized by the entire clinical community also. So we are complying like with the latest clinical guidelines as well as the regulatory guidelines. So you have the safety of mind that our assays are biotin interference free. So these are like the system designed for the smallest analyzer, Shine 1000. You have the integrated display, reagents, samples, and on the side like you have the trigger A and the trigger B, which are like the substrates for the actinium ester. But you can go for different labels like the API, alkaline phosphatase, or HRP. And the side like it shows you like the barcode reader that you can interface. And at the back you have the inlet and the connectivity like for the waste. Similarly, for our medium throughput Shine 2000, you have the reagents, and as you can see there is a sample rack. So there are 6 racks of 10 samples each, and you have a dedicated waste, as well as like for the substrates you have a special containment in the front, and in the side you have the same connectivity like to the LAN through the barcode reader, and there is also like an integrated internal barcode reader also. And then you have the liquid inlet and the outlet at the back. Now some of the salient features like of our analyzers are: we have non-touch vortex mixing, we have 3-phase magnetic washing, so which enables you like to go for very stringent washing steps. Then you have like assay cups that are single, so you just put the assay cup within a particular containment and the system will align all by itself. So rest, please refer like to our webinar where we have discussed all these technologies in more depth. Then we have like a dispensing probe which is made of a particular grade of the stainless steel, which is highly resistance, contamination free, and you can detect the clot, air bubble, and the liquid level. The washing of the probe is done via 3 water jets. One is coming from inside the dispensing needle and another 2 are coming from the sides. So there is no carryover from one assay to the another. Then you have a photomultiplier tube which has a wide range like for the readout of the assay signal. You have the same assay cartridge or the reagent cartridge which you can use like for the different throughput of the assays, and all our analyzers are using the same cartridge. There is a sample rack that can contain like the samples. Then you have like the substrate containment and the waste containment, which is like for the trigger A and the trigger B for the actinium ester, but other substrates for the other labels that we are using. Now the latest one that we launched at the AACC last year is the Shine 8000 or 9000. This has 2 reagent trays. This is a standalone system, and the capability of the instrument is it can go like up to 900 tests in a single module. But if you combine 4 modules, then it is 3600 tests. So the maximum sample capacity is 410. 300 are regular samples, another 100 they come as a catch, and 10 you can go like for the stat module. So there is a double reagent needle mechanism which is proprietary to Fapon, and there is a multi-stage magnetic washing. It involves like the RFID scanning for scanning like the barcode like of the assay cartridges, and there is a barcode scanner like for the patient samples. Then we have the system, all these analyzers can run like all types of the clinical samples, and nearly all the commonly used sample tubes, they can be used for the different analyzers. So you can use the analyzers for practically all types of the clinical assays. Then we have a highly diverse CLIA menu for the different diseases, and Fapon has like the most extensive, you can say, like the menu for the tumors, and many of our assays are acting as a state of the art for the entire industry. As an example, like for the infectious disease and the gastro panel, we have the HIV combo, HCV, then Treponema palladium, so which has very high performance in the entire industry. Then for the PG1, like Pepsinogen 1 and the Pepsinogen 2, if we compare ourselves with the leader in the market, we have a very high correlation. So most of our customers tell us like we have like one of the best performances in the entire industry for the different assay panels. So we recommend like the use of our dedicated reagents by the entire IVD and the clinical community. Then we have like a unique and proprietary protein-free blocker, which is chemically synthesized. The size is smaller, like 0.72 nanometer, and the molecular weight is 4 kilodalton. So you can say like it is around 15 times smaller than the PSA, and because of the smaller size, it can block the empty sites on your magnetic beads after the coating of the biomolecules, and this leads like to the more effective blocking, and we can produce it at a very high manufacturing capability. So you can expect very quick manufacturing, and it leads to high signal to noise, decreased NSP, high production yield. And we have it like for all the multifarious assay format, whether it is like CLIA, ELISA, rapid test, point of care, microfluidic, or the multiplex test. There is another webinar like at the AACC that we did last year, which is specially dedicated to protein-free blockers and the heterophilic antibody blockers. So we are providing the link in the webinar here. Please go through it and get back to us if you have any query. Regarding the protein-free blocker, it is composed of hydrophilic and hydrophobic chains, and we can use it as a blocking reagents together with the, like after the coating of the biomolecules to your magnetic beads, you can block the empty sites in a subsequent step. Or you can already use it like in your capture reagent as well as the detection reagent. Capture reagent with the magnetic bead and the detection reagent with your CLIA label like the acridinium ester, where it will give you highly effective blocking. Now Fapon has like an extensive variety of the active and the passive blockers for the heterophilic antibody. As you already know, like the heterophilic antibody interference is one of the key issues in the clinical community, and all the IVD manufacturers as well as the clinics want to get rid of the heterophilic antibody interference. But once you launch the assays, then the possibility for counteracting the interference is very less. So these particular blockers are intended for the IVD manufacturers that they can use it for the dedicated formats, and we have a large variety. So you can approach us for guidance which blockers will be ideal like for your specific application. Most of the large industrial giants, small and the medium players in the entire industry, they are using like our bulk reagents for the CLIA, for ELISA, for the lateral flow, and we are assisting them like at different stages. So in some cases we provide them like the raw materials, we provide them with the reagents, and we assist them in the protocols, clinical evaluation, correlation with the predicate, which particular blockers they can use. Sometimes we also do like the labeling and the conjugation for them. And at other times we complete like the optimization and help them like in arriving at the assays very quickly. Sometimes the different immunoassay formats also plays a role, so we also enable them like in arriving at their particular optimized assay really quickly, because we are producing the raw materials, reagents, instrument, and the protocols for a large variety of the assays. So we have extensive experience in this particular field, and whenever we produce like the raw materials and the reagents, it is always the case that we have to compare ourselves with the best in the industry as our predicates. So we have extensive experience, we have the established key opinion leaders that guide us at every stage of the development. So please get in touch with us if Fapon can assist you or empower you in the development of the particular assays. And we are always interested in the new applications, new biomarkers, new technologies, where we want to work together in cooperation with you. Now regarding the Fapon raw materials, we have a rich menu for the point of care testing for the infectious disease, inflammation, cardiac biomarkers, for the respiratory panel, hormones, gastrointestinal, and then we have a large variety like for the drugs, and some other panels like the torch, HbA1c, TSH, vitamin D. So you can use our dedicated point of care testing menu for the development of rapid test and the fluorescent immunoassays. And if you compare ourselves in terms of the clinical significance, performance, you can see like for HIV, HCV and the Treponema palladium, most of our customers, they have already achieved the WHO PQ using our raw materials. And our HIV fourth generation, it can detect up to HIV type 2, and the sensitivity is very high in comparison to the leading predicate in the market. Similarly, for the HCV and the Treponema palladium, we are highly cost effective, and our customers and the entire market tell us like that we have one of the best performances in the entire industry. So please feel free like to get back to us if you want like more data pertaining to this particular raw material, so we can share our experience and our findings and results with you. With regard to the tropical diseases like malaria and dengue, so there also Fapon is known as a leader in the entire industry, and most of the raw materials that our customers are using for the development of the test, whether it is for malaria in terms of the lactate dehydrogenase, where many of the customers already achieved the WHO PQ, and for the dengue, we have the highest market share, the prices are affordable, and the best part is like we have different base pairs of the antigen and antibody, so Fapon can guide you which particular base pairs will be suitable for your particular assays, and this will help you in the development of the assays very quickly, because screening of the raw materials, going for the optimized protocol, consumes most of the time for the IVD manufacturers. But using the assistance from Fapon can empower you to develop this test really quickly. And for the cardiac troponin I, anti-proBNP and HbA1c, our assays have been used a lot by the entire industry. We have like the outstanding sensitivity, specificity, as well as like the very high stability and correlation with the gold standards in the entire industry. And as I mentioned, like you can get in touch with us where we can share like the findings and the results with you. From our experience, it will help you in the development of these particular panels in your particular format really quickly. Our Helicobacter pylori has much wider acceptance, and it has the best sensitivity because we are using like the multi-gen antigenic epitopes. And you can develop the assays like based on the particular base pairs that you are interested in, and the performance of the assay is really good, which we hear from the entire industry. Then Fapon is also providing like the biochemistry raw material solutions. We are providing you like for the CRP, ASO, D-dimer, rheumatoid factor, cystatin C, and the different base pairs. So this helps you like in the development of the assays quickly. And we recommend to our customers which particular antibodies and the antigens they can use, and what latex particles and the coupling procedures they can employ for the development of their test. So one of the key features like of our HbA1c is you don't require like a second antibody as used like in the conventional test, because the antigen molecules like hemoglobin A1c, they can bind to your latex particle via physical adsorption. After that, the antibody can directly bind, and we are employing like a single internally cross-linked antibody, so you don't need like a complex reaction procedures where you require like the second antibody, and you have like the best linearity, sensitivity, precision, correlation with the leading predicate, and the assay is highly stable. So you can get back to us if you need like more information about any of the raw materials which we have discussed today with you. And if you want to know more like about the Fapon IVD products, solutions, our future direction, and the ecosystem cooperation that we are forging with the IVD manufacturers and the clinical community, please do visit us at MedLab Middle East in Dubai. Thank you very much. Any queries, please?